Okay, good morning, friends. Now it is the time for us to start because the time is very short and the syllabus that has to be completed is very large. So that's why we cannot waste any time. So straight away we'll start the lecture or online class. In 6th sem, one of the electives is plant disease management, and we will learn about various plant diseases and the methods of their management in 6 classes. See, as you know, we have a class today, that is Friday, tomorrow Saturday, and day after tomorrow, that is Sunday. We have 3 classes. Similarly, we have 3 more classes on the next Friday, Saturday, and Sunday. So altogether, we have 6 classes allotted to complete this plant disease management. As you know, now this syllabus of plant disease management, it comprises, 4 blocks are there, and in each block there are 3 units, and that way there are 12 units that are to be completed in 6 classes. So hence, I think it is better to complete 2 units in each class so that we'll be in a position to complete all the 12 units in the allotted 6 classes. So now, in the block 1, there are 3 units, and of the 3 units, we will try to complete the first 2 units, that is fungal diseases and fungal diseases 1 and the fungal diseases 2. So this tells you that, fungal diseases form, major part, or fungal diseases cause a lot of damage to the crop plants when compared to all other pathogens, that is bacteria, viruses, mycoplasma, or, animal pathogens, including some protozoans or insects or, borers, etc. combined together also do not equal to the number of pathogens that are present in fungi and the magnitude of destruction these fungal diseases cause. So now, the entire syllabus, if you look at the syllabus, generally we have what we say in block 1 and 2, we discuss about the plant diseases. That is, diseases caused by fungi, diseases caused by bacteria, diseases caused by viruses, and diseases caused by mycoplasma. In addition to this, we'll also discuss about various pests that are common to cotton, pests that are common to paddy, not only in the fields, but also in the storage areas. That is, even the godowns, a lot of damage is caused by the infestation of these storage products with pests. In addition to this, we also have what we call the parasitic plants. They also included under pests category. They too cause a lot of damage when infest our crop plants. So this block 1 and 2, mainly they concentrate on various plant diseases. Generally, plant diseases, we confine ourselves only to crop plants. Say in 5th sem, you read about crop production, wherein we discussed about various crops. Your cereal, that is food crops we have seen, fiber crops we have seen, and the cash crops we have seen, and plantation crops, etc. we have seen. So we have selected a few important crop plants, and we'll discuss about the major pathogens that cause loss to various crop plants. So this block 1 and 2, as I told you, discusses about the pathogens of, that is, plant pathogens is better. So plant pathogens, that is fungi, bacteria, viruses. mycoplasma. Then your pests, that is animals, that is various pests that cause a lot of damage to cotton and paddy. So both block 1 and 2, we discuss about various pests and diseases. We discuss about various pests and diseases in block 1 and 2. And in block 3 and 4, we'll discuss about how to manage these pests, that is pest management. How to check, how to protect our crop plants from the infestation or from the attack of these plant pathogens and also animal pests. So that way, we'll discuss about the various pathogens and pests in block 1 and 2. And in 3 and 4, we'll discuss about various managerial practices to check the spread of or the infestation of these pests and how best we can protect our crop plants. This is what we call management of these pests and diseases. So straight away, we will move to various plant diseases. Now, generally, we will discuss about fungal diseases and mostly fungal diseases. So that is fungal diseases that are present in Unit 1 and also in Unit 2, we'll try to complete because time is very short and as I already told you, the syllabus to be covered is very large. So we'll be moving a bit fast. So fungal diseases, as you know, fungi, as you know, now in Whitaker's classification, because of their specificity, they have been included in a specialized kingdom of their own. And these fungi differ from other crop plants, or other, plants because they are heterotrophic. They are heterotrophic. They live either as parasites or as saprophytes. They are achlorophyllous. So generally, whenever we say plants, generally plants are autotrophic. But these fungi are completely, they are heterotrophs. Maybe living as saprophytes or as parasites. So now, in Unit 1 and 2, we'll discuss about, various fungal diseases. So generally, fungi produce a large number of diseases in almost all the crop plants. And when they infect a given crop plant, they kill or damage the tissue or the cells, and thereby they produce certain symptoms. Now, these symptoms could be in the form of what we call necrotic symptoms, or may involve what we call hypertrophy or hyperplasia or wilts or damping off or soft rot, etc. are some of the symptoms that fungi cause when they infect a given crop plant. So generally, we will, in the syllabus, generally in fungal diseases Unit 1, we will discuss about various important fungal diseases that are common in our staple crop plants. That is rice. We'll discuss about 3 important diseases caused by fungi in rice, and also 2 important diseases caused by fungi in another staple food, that is wheat. So we will begin with the fungal diseases. Now, before that, let us know something about what we call the meaning of a disease. What do you mean by the meaning of a disease? Now, as the term tells you, dis-ease. Ease means when you are comfortable, when you are uncomfortable, when you face a discomfort, that we call any deviation, any disturbance from a healthy condition, interfering with the normal structure or physiological functions. If it impairs either your normal structure or your physiological functions, then we can say that that particular organism is unwell or diseased. So now this diseases, generally I told you, it involves in either killing of the cells or the tissues, thereby subject the plant to a kind of stress. So under stressful conditions, under dead cells or tissues, the crop shows certain symptoms and its yield gets affected. Not only yield, not only the quality, but even the quantity. So both qualitative and quantitative degeneration will be there when a pathogen, when a fungal pathogen infects a given plant. So now, generally, we before discussing about various fungal diseases, let us discuss about few important terms that are very commonly used in discussing about these various plant diseases and their management. So now, generally, the symptoms, symptoms are nothing but external signs that are exhibited by an unhealthy plant is what we call a symptom. Now, generally, the symptoms are of two types. One we call, systemic or localized. These symptoms may be systemic, or the disease could be what we call a systemic or a localized. Systemic means the pathogen or the disease symptoms are found throughout the plant body, starting from stem tip to root tip. If that is the case, we call a systemic. Localized means the symptoms are restricted to only a given area. Such we call localized. Now, the other common terms we get while discussing is what we call host and parasite. Host and parasite. The parasite is the pathogen or the causative organism that lives and causes damage or it draws its food from the host. And the host is nothing but that provides shelter, that gives nourishment to the parasite that is depending on it. That is the host. This is the parasite. But you have to remember that all parasites may not cause diseases. Most of the parasites generally they cause diseases, but this is not a general phenomenon. There are some cases wherein even a pathogen, a parasite may not cause a disease, but of course it draws its nourishment from the host. Thereby, the host is subjected to some loss. And there is another term what we call a pathogen. So pathogen is a disease-causing organism. It certainly causes some disease or discomfort in the host. So now, generally, another term is what we call inoculum. Inoculum is disease-causing organism is known as inoculum. Inoculum could be a fungal spore, it could be a bacterial spore, it could be a virus particle. So that has the potency to cause disease in a host that we call inoculum. So now the other is infection. So infection occurs with the penetration of the pathogen, disease-causing organism into the tissue. Generally, the entry into the host will be generally through epidermis or through the epidermal pores, what we call stomata, that are mostly found on, leaf. When compared to other, generally foliage. Whenever you say plant, what comes to your mind, that is green foliage. Leaf will be there. So leaf is the thing, that, absorbs solar light, that is radiation. And it is the organ where photosynthesis takes place. So generally, the infection or entry, penetration will be through leaves, generally through epidermis or through stomata. But rare cases, there are certain cases wherein, for example, in case of foolish seedling growth, a Botany disease, wherein Gibberella fusicori or Fusarium fusicori, wherein the conidia make an entry into the plant body through roots or root hairs. Then another important term, generally these are the terms, generally you get in your objective. So incubation period. So the time lapse, that is the time interval between the entry of the inoculum and appearance of the symptoms. This duration, what we call incubation period. So with this brief definition of terms, we now will go to the diseases. So generally, another important thing you have to know is the diseases. Generally diseases are infectious diseases and non-infectious diseases, or they are otherwise called abiotic diseases and biotic. Abiotic, as the term tells you, these are not infectious. These are not, parasitic. Abiotic, there is no living organism is involved. Hence, it cannot be an infectious. It cannot be in a position to transmit from one plant or from one organism into other organism. There is no inoculum. So here, generally, because of the inclement weather conditions. So mostly because of inclement weather, unfavorable weather conditions, generally plants do suffer certain discomforts. We call such discomforts with abiotic forces as abiotic diseases. Generally, under this, generally temperature, your moisture is there. Generally, your soil nutrition will be there. So our maximum rainfall could be there, or what we call snowfall will be there. These are all inclement, not favorable. So say, for example, if temperature is very high, there is a burn. The leaves get burned, or the coleoptile or the stem tip or the branch tip may get burnt. Or if the moisture is too much, then that is also not good for the plant growth or for plant's health. So even if the nutrition, of course, we have you read about in your plant physiology, nutrition we have seen, wherein plant nutrition, we have macronutrients, micronutrients, etc. If the balanced diet to our human beings, so also if the macro and macronutrients are not present in required quantity in the soil, plant do suffer from certain diseases or symptoms we have seen. So all these are not contagious, not infectious. So hence they fall under what we call abiotic. So one or two examples, so some, under your, objective, you may get it. Which of the following is a abiotic disease? For example, black heart of potatoes. Black heart. So if you cut the potato, in the center, you will see blackening. So that what we call a black heart of potatoes is there, and tip burn of lettuce, a tip burn of palak we have seen. So then heat canker of flax and spinach tip burn. These are some of the examples for what we call abiotic diseases. Of course, then biotic diseases, I already told you, biota, that is living organisms causing diseases in crop plants. It could be a fungus, it could be a bacterium, it could be a mycoplasma, it could be a virus. Of course, it could be even through nematodes. We know nematodes, they infest, or even certain protozoans, certain borers. Say, for example, rhizome borer of turmeric or ginger, etc. are there. They bore into the rhizome, they eat away, and they cause a lot of damage. So these are fungi, bacteria, mycoplasma, viruses. They cause a lot of damage to the crop plants. So we concentrate on only these. That is fungal diseases we study. We study bacterial diseases, mycoplasma diseases, and viral diseases. We confine ourselves. So now we begin with, there are three important, diseases. There are three important plant diseases, especially confined to what we call rice. So rice, is the staple food, as you know. So more than 60% of the population in the world depend on rice as their staple food. The most populous countries of the world, that is, of course, India and China, and South Asian nations where the staple food is rice. So hence, more than 60% of the population in the world depend on rice. So now this important rice plant or rice crop suffers from few fungal diseases. So there are three important, fungal diseases in our syllabus. One is what we call, blast or paddy blast we have, and the brown spot of paddy we have. And the last one is what we call, foolish seedling growth of rice. These are the three diseases that are there in our syllabus. Now, the first one is the paddy blast. Why the term blast? Now generally this paddy blast, it is caused by what we call Pyricularia oryzae. Of course, new name is Magna porte oryzae. Magna porte oryzae is the new name, and Magna porte grisea, Magna porte oryzae. This is the perfect stage, and our Pyricularia oryzae is the conidial stage. So now this Pyricularia oryzae, a fungal imperfecti. So now whenever its sexual stage or perfect stage is discovered, it would be transmitted or transferred to either Ascomycotina or Basidiomycotina, as the case may be, based on the perfect spore they produce in their life cycle. So this paddy blast, it is caused by Pyricularia oryzae or Magna porte oryzae. Now, why the term blast means whenever the fungal spores infect the plant, generally they infect or they discolorize, they damage the leaf lamina, leaf base, even the stem part, the nodal part. So every aerial part of the plant body, including your panicle, your glooms, your neck of the panicle, every part of the plant body becomes dried and brownish. And if you look at that, that diseased part, it looks as if it is burnt. Hence the term blast. It exhibits a burnt symptom. Hence the term blast. As it is occurring on paddy, hence we call paddy blast. So now the symptoms, if you look at the symptoms caused by the paddy blast by Magna porte oryzae, generally the symptoms will appear, pale, that is whitish or creamy pale, spindle-shaped structures will be there, and generally they will be surrounded by a brownish halo spaces, and generally these small symptoms, spindle-shaped symptoms, in due course they coalesce, they fuse with one another, and in due course the entire leaf lamina becomes dried and becomes or exhibits a blasted appearance. So hence the blast. Now, if so, disease symptoms could be from a seedling stage. If the symptoms appear at the later stage, that is, if they occur when the plant is in the panicle or in the milky stage, if that is the case, generally you will see choppy seeds. Seeds will be there, but no endosperm, no embryo will be. Choppy seeds will be. So because of this, based on the severity, the loss could be varies from 70 to 90%. Now, generally, the organism, that is Pyricularia oryzae or Magna porte oryzae, now this, it mainly produces by a production of conidia. So generally, these conidia are born on the conidiophores, and these conidiophores come out through stomata, and the conidia are generally obclovate or pear-shaped structures, 3 to 4 celled structures, and they are produced in large numbers. They are produced in a single night, from a given spot, you will see production of will be ranging from 40 to 60,000. Such a large number of spores are produced, and when they are produced and when they get detached, and when the conditions are moist and warm and moist conditions are there, they get blown away by the blowing wind, and they fall. When they fall on the fresh, healthy leaf, this conidium germinates by a germ tube, and either it penetrates through epidermis or enters through stomata, and within no time, it enters and it colonizes the entire mycelium. It ramifies in the intercellular space of the leaf tissue, and it ramifies. And again, you will see conidiophores coming and production of conidia at the end. Generally, at the end of the season, generally the fungus enters into what we call sexual stage, and in the sexual stage, generally they produce what we call sexual fruit bodies, what we call perithecia, a flask-shaped structure known as perithecium into the because it is ascomycete. It produces many asci. Within the ascus, you will see production of ascospores. So these ascospores are the sexual spores. So now this is how it causes a lot of damage to the paddy crop, and how to check. Generally, the control measures are categorized into 3 or 4 categories. One is cultural method, the chemical method, and the physical method are there. And generally, we go for chemical method only as a last resort. Generally, we go for cultural method, then we go for physical methods. Now, generally, in cultural methods means generally, we see that the pathogen does not enter our crop. We go for, crop rotation, and we see that the collateral host, generally grasses, the other, rice-like plants which grow along with the rice in the rice field, they are to be, they form the reservoirs. When you cut, when you harvest your, paddy, generally on the hedges, on the bunds, or on the channels, you will see many grasses. Now, on that, the pathogen perinates, or even after harvest, the infested, the stubules or the fallen leaves, they form the, they perinate there. They over summer there. They over winter there. When you again go for transplantation of the rice, your rice plant is prone for infection. So hence, what should we do? You should go for crop rotation, or because the pathogen can be even on the blooms or even within the seeds. Hence, the seed before sowing should be soaked in the water for a few hours. Then it should be spread on the sunlight for a few hours, or they have, a lot number of research has been done. So if it is, if you can put the seed before sowing, at 45 to 50 degrees centigrade for 10 minutes or so. Later on, you spread on the floor. So during this time, the pathogen which is found, if it is seed borne, it germinates. And when you spread it under sunlight, the tender hyphae, they die, so that your seed becomes pathogen free. So by sanitation, that is removal of all the stubules and the fallen leaves, and removal of all collateral hosts, etc., should be done. Then if it is not possible, then you go for at a last resort, go for chemical. So generally, seed treatment can be agraston, serrasan, a dry, dry matter treatment. That is these powders, agraston, serrasan, or you can go for what we call wet treatment by going for copper sulfate or calcium hypochlorite, etc., can also be used, or phenyl mercuric acetate, etc., can be used, or you can go for what we call copper fungicide or phenyl mercuric acetate spraying. If you find these symptoms on the foliage, then you go for the spraying, foliar spray. So by doing all these things, we can protect our. Then the second one is brown leaf spot. Brown leaf spot is another fatal disease found on the rice crop. Generally, it is found on almost all the rice growing areas. And this brown spot is otherwise known as helminthosporiasis. This is caused by Helminthosporium vorizae. And of course, this Helminthosporium vorizae, Dressler vorizae is the new name or the perfect stage of this fungus. And now, there is a history, in 1942-43, with this Helminthosporium vorizae or brown spot of rice, caused a lot of damage. Entire 90% of the crop in Bengal. In 1942, of course, Bengal was East Bengal, that is nowadays what we call Bangladesh, and the West Bengal, East Bengal were united, and then that is an area where crores of people lived, and because of the total crop failure, an estimated 50 lakh people died. Hence, it is called what we call Bengal famine. Bengal famine. Under short note you may get because of which of the following diseases Bengal famine occurred. That is Helminthosporiasis or Helminthosporium vorizae or Dressler vorizae that causes black or brown spot of rice. Now here also, it also produces by conidia, and of course here the conidia are a little bit big when compared to blast of rice. Now here the conidia are big, and they are what we call clavate or sickle shaped structures, and they are 5 to 10 celled structures, and they are produced in large numbers. And of course, the pathogen survives in collateral gramineous plants when rice is not available, especially this Dressler vorizae. It over summers, or it survives in gramineous plants such as Lysera hexandra, Echinocloa colonum, or Citeria italica, or Cynodon dactylum, or these are some of the gramineous members which grow along with the rice crops, and they act as collateral hosts. When rice is not available, these survives. The pathogen survives on these hosts, and whenever you again plant rice, from here they get transmitted onto your healthy rice crop, thereby cause a lot of damage. So what you remember here, the brown spot of rice is caused by Helminthosporium vorizae, or otherwise known as Dressler vorizae, and this is the cause for what we call Bengal famine. And generally, its perfect stage is Cochleobolus meavinus. Cochleobolus meavinus is the perfect stage of what we call Dressler vorizae or Helminthosporium vorizae. Here, the fruit body is what we call a perithecial fruit body. And in this, the perithecial is it is like this, and it has an opening, what we call ostiole, and the swollen part bears a number of asci, and each ascus bears a number of 8 ascospores are produced. And when they mature, generally, I told you have to remember. So generally, when the conditions are favorable, fungus reproduces by asexual means. Where at the end of the season, when the rice comes for maturity, at that time, generally you will see the fungus entering into sexual stage and producing through what we call ascospores. And these ascospores, they remain dormant, and when conditions are favorable, they germinate by a germ tube, and thus make an entry into the plant and produces mycelium, and within no time, the monokaryotic mycelium becomes dikaryotic mycelium, and this dikaryotic mycelium produces what we call by conidia or by through ascus. So now here also the how to check or control the diseases by eradication of the collateral host or by cleaning and burning of the previous infected plant debris, and generally the leaching of the metals. Generally, iron, manganese, and potassium are found to be very, very important. If the plant, if the soil suffers from the deficiency of these, plant is prone for, or the crop is prone for this brown spot disease. Now, this spacing, the proper spacing should be there between one hill and the other hill. Proper spacing should be there. And the nitrogen fertilizers, too much nitrogen fertilizer should not be given. And if they are given, they are to be given in split doses. And generally, of course, before sowing, generally hot water treatment is necessary if the pathogen or the conidia are present on the, there is every possibility, seed-borne, because it is a seed-borne. So hot water treatment, keep the seed for 10 minutes in 55 degrees centigrade and then spread it on the floor under sunlight. So within 2 or 3 hours, whatever the mycelium, the spores germinate, they get killed. So then chemical control will be dry dressing. That is a dry powder such as Serasan, Agrasan can be used, and wet dressing by using what we call silver nitrate or copper sulfate or mercury chloride or formaldehyde, etc. can be used. Or if you want to go for foliar spray, generally go for fungicides such as dithanium 45 or dithane 78, or you can go for what we call Borodax mixture spray or Blitox. Some of these sprays can be used. And the best way is to go for these disease-resistant varieties such as IR24 or Padma. These are found to be quite resistant for this brown spot disease. Okay, then the other important fungal disease, what we call Botany disease. Botany means foolish seedling growth. Foolish seedling growth, that is, this is caused by Fusarium fusiculae. The previous name was, Gibberella fusiculae or Fusarium fusiculae are one and the same. And this was, and the seedling, generally the fungus infects the young plants, that is young seedlings. Now, if the seedling gets infested, the microconidia, if they make an entry through root system, and within no time they ramify, produce lot of mycelium, and this mycelium, this Fusarium fusiculae or Gibberella fusiculae produces a growth hormone known as gibberellic acid. Under the influence of gibberellic acid, the plant shows overgrowth. So when compared to the healthy plants, these infested plants show double the length of the other healthy plants. So this overgrowth is what we call foolish seedling. In Japan, generally, it was first discovered in Japan and by what we call Shotaro Hori. They may ask you under short notes, who was the scientist, who was the man associated with the discovery of this foolish seedling growth. So Shotaro Hori, a Japanese man, is credited with the discovery of this foolish seedling growth. And of course, the cause was identified later, and it was Gibberella fusiculae, and which produces what we call gibberellic acid. That is the reason for the overgrowth of the seedling. So as the seedling is growing over and over, hence we call a foolish seedling. A foolish seedling. And this overgrowth results, this overgrowth, results in this overgrowth, the plant that is Fusarium produces in the meanwhile a number of conidia, macroconidia and microconidia, and these macroconidia and microconidia enter into the conducting tissue, and thereby it causes a kind of wilt. So the seedling which grows, it starts drying. It gets dried. So and this micro and macroconidia, they remain dormant in the soil, and whenever you go for fresh, sowing, then they make an entry into the plant, and thereby producing what we call foolish seedling growth. So the infected plants, generally the infected, because they are going tall, generally they are tall, they are thin, they show, they give, less number of tillers, and the leaves are not dark green, they become pale green, and within few weeks, the leaves start drying. They get dried and hence the plant dries. And there is no question of bearing any grains. Even if it survives, even this overgrown, because the plant is overgrown and its yield will be very less. So even if it comes to flowering and fruiting, even the number of seeds from the panicle will be very, very less. So now here the pathogen, this pathogen, what we call Fusarium physicori or Fusarium moniliforme, I told you, reproduces by what we call micro and macroconidia, and its perfect stage is Gibberella physicori, and it leaves this Fusarium moniliforme or Fusarium physicori leaves. It's an obligate, it is a facultative parasite, and it lives both as a parasite and as also saprophyte. So elimination of the affected plant parts and treating with agrasone cyrazone or using 1% copper sulfate or formaldehyde or carbendism, etc., carbendism spraying, etc., can be undertaken to check this. Or foliar spray generally by using what we call benomyl or carbendism, etc., can be sprayed. Then we move to what we call wheat rust. You are acquainted with this rust disease. Now, this rust, as you know, why it is known as rust, So because the pustules or the streaks found on the streaks or the pustules found on the plant, they are rusty in nature. Hence, it is now called a rust, as you know. And this rust, it causes a lot of damage. And the pathogen is what we call Puccinia graminis tritici. Puccinia causes this rust disease in wheat, and it causes a lot of damage to the crop. And generally, this rust, this in the rust or the streaks or the pustules, you will find spores known as uridospores. And the sorus is known as, if you take a section of that pustule or the infected part, you will see accumulate single-celled binucleate spores called as uridospores. And this Puccinia, is what we call a macrocyclic heteroecious fungi. Heteroecious means it needs what we call two hosts. Wheat is what we call a primary host, and barbaris is what we call a secondary host. So wheat is the primary host and the barbaris is the secondary host. Now, hence it is called heteroecious. It requires two hosts for the completion of its life cycle. And it is otherwise macrocyclic. It produces, in its life cycle, it produces 5 different types of spores in its life cycle. One is pycniospores, ichthyospores, uridospores, teletospores, and finally basidiospores. So like this, as 5 types of spores are produced in its life cycle, hence it is called a macrocyclic heteroecious fungi. So generally, on the barbaris leaf, you will find 2 stages. What is pycnial stage and ecial stage are found, whereas in wheat, generally, we find what we call uridospores. And the last, at the end of the season, generally, uridospori generally change to what we call teletospores. Now, at the end of the season, generally, so when the season, generally the uridospores are rampant. They're produced in lakhs and lakhs, and they are even they cause what we call secondary infection too. So at the end of the season, the uridospori changes to what we call teletospores, and these teletospores are bullet-shaped structures, and they are 2-celled structures. They are two cell structures, and generally they are black in color. And generally they are found, these streaks or lesions are found even on the stem. As the symptoms are found on the stem, as they are black in color, hence this is also known as stem rust or black stem. Black stem. As the pustules are black in color, as they are confined to stem, hence we call black stem. So iridospores, generally they are confined to leaf areas, and generally they are rusty in nature. So this causes a lot of damage to the crop, and generally the best way to control, this is what we call going for a cultivation of resistant varieties. We should go for resistant varieties, and you should go for fungicides to control this. Generally, you can use sulfur 25 to 30 pounds per acre can be used, and of course certain diethane and zinc sulfate, etc., can be used as effective foliar spray to control this rust disease in wheat. And on wheat, we also have another disease, important disease, what we call smut disease. Now, generally, the rust, rusts are red, rusty in nature. Smut means which are black in nature. So, smut of wheat. And here, this is what we call loose smut of wheat. So, it is caused by, generally, smuts are caused by Ustilago, Chinus Ustilago. And generally, Ustilago triticea was the old name. Now, this is Ustilagus sagittum is the new name of this loose smut of wheat. Now, generally, the perfect spores, generally are what we call chlamydospores are produced, which are black and sooty. And generally, these spores, they're very light, and they get disseminated through air, and they fall on the panicle, ear or the panicle, and they fall on the glooms, and ultimately they make an entry through stigma, and they enter into the ovary part. And in the ovary part, after their entry, the chlamydospore or the spore germinates and produces a epibasidium and a four-celled structure. And from that four-cell structure, you see the formation of basidiospores. And these basidiospores, and they fuse. Two different strains of these basidiospores. They fuse, and these monokaryotic mycelium becomes dikaryotic, and that way it enters into the major part, that is which ramifies and replaces the entire ovary with its black sooty spores. And the diseased, entire the ear, all the kernels get replaced by black spots. And when there is a gust of wind, these black suits are blown away, and you will see the empty rachis. The empty rachis, because no seed formation is there. So, so here what you have to see for all these things, what you have to remember is name of the disease and the pathogen, and the perfect stage and the asexual stage, and the mode of entry and the method of control. These are the few points you have to concentrate while answering the various questions. Now, this is about the fungal diseases, important fungal diseases of rice and wheat. Then we move to fungal diseases. Next one, in next fungal diseases, we will discuss about what we call, here we have, under this we have, smut of sorghum is there. We have, that is, smut of sorghum is there, and green ear of bajra leaf is there, and powdery mildews of cucurbits we have in syllabus, downy mildew of grapes we have, and corn smut we have, and leaf spot of groundnut we have, and last one is wilt of cotton we have. So, say to start with, smut of sorghum. Now the smut, I told you rust and smut. Rust are rusty, smuts are blacky. Now the smut of sorghum, the pathogen is what we call Sphacilotheca sorghum. Now this in sorghum, we have three important species of Sphacilotheca. Now one is what we call grain smut or covered smut. This is caused by Sphacilotheca sorghum. And the second one is loose smut of sorghum. This is caused by Sphacilotheca cruenta. And the last one is head smut of sorghum, head smut of jowar. Head smut of jowar is caused by what we call Sphacilotheca reliana. So that way, sorghum or our jowar, sorghum or jowar is caused by grain smut, loose smut, and head smut, caused by three different species of Sphacilotheca. So generally, this disease is generally, the spores, that is the chlamydospores or the smut spores are seen clinging to the seeds. Hence, seed treatment is very, very important. The hot water treatment or treating the seed with, copper sulfate or what we call silver nitrate, etc., or formaldehyde, etc., can be used. And best thing is to remove all the collateral hosts and to clean sanitation should be there. Go for what we call crop rotation and use what we call disease resistant varieties, CHS5, CHS8, etc. are there, which are very effective in facing this smut. The second one is what we call bajra, this bajra, what we call pearl millet, and this fungus is what we call Sclerospora graminicola. Why it is known as green ear, if you observe the infected leaf, the glooms or the stamens, etc., they become leafy in nature. If you look at the infected, ear, upper part of the 1/3 of the upper part of the ear, it becomes green leafy. Hence the name green ear disease of Bajra. And the infected, this pathogen at the later stage, when it enters into the ear, it transforms the ear into leafy structures. And when it is in younger stage, when in the growing stage, the leaf, on the leaf, you will find many pustules. Generally, on the upper surface, you will find the symptoms begin with oily, pale, green spots will be there. And from the undersurface, through the stomata, you will see white cottony outgrowth. White cottony outgrowth will be there. And these white cottony outgrowths are nothing but they are sporangiophores and which bear hundreds of sporangia. It is a Phycomycotina species, and these sporangia they germinate into what we call a biflagellar zoospores. It swims in the water drop that is present, and of course later on it it loses its flagella and it germinates through germ tube and makes an entry into the host, and after entering it ramifies and it produces through conidiophores. So these conidiophores are seen coming through stomata and producing conidia in large number. The conidia are pear-shaped structures and produce in large numbers. They get disseminated and form the second inoculum. So now the third one is what we call powdery mildews. The powdery mildews, as the name indicates, the powdery mildews, if you look at the infected leaf, generally powdery mildews are cucurbits. Generally in cucurbita members, in all cucurbits, in pumpkin or your bottle gourd, etc., or even in grams, especially in green gram, black gram, on the leaves, you will see as if somebody has put some powder. Somebody has put it powder, but no powder from outside. But this fungus, Erysife sicaracearum, produces by producing a number of conidia, otherwise known as oidia or conidia. The conidia are produced in such large number, millions of conidia are produced. And these conidia, even under dry conditions also, they have the ability to germinate and produce the mycelium. Again, they enter into reproduction phase and produce through asexual means by producing number of conidia. So all these conidia are produced excessively, and these excessively produced conidia are seen spread on the surface of the leaf and showing powder. So hence the name powdery mildews. Then downy mildew, this is a very important thing. The downy mildew of grapes. In the downy mildew of grapes, the pathogen is Plasmopara viticola. Vitis vinifera is the scientific name of grape. So from that, this Plasmopara viticola causes downy mildew of what we call grapes. So on the grapes, infested grapes, the leaf, generally the tender parts, even the young leaves or the petioles, or even the other young parts, of the plant body get infected by this Plasmopara viticola, and it reproduces similar to our bajra, wherein it reproduces by producing what we call a lax of sporangiophores, and ending with a number of sporangia. So these sporangia come out from the lower surface of the leaf. So generally you will see on the upper surface pale and generally light green pustules or spots will be there. So as the disease progresses from the lower surface of the leaf, you will see production of a large number of conidiophores and bearing conidia. And these conidiophores, they show what we call cotton-like, as if cottony outgrowth will be there. Hence the name downy mildew. So now, not only on the leaves, but even on the inflorescence stalk also, even on the grapes also, you will see the growth of these downy structures. So when the inflorescence gets infested with this, the entire, bunch of the grape may get detached, may die, may get rotten. So hence, a lot of damage will be there. And, this, especially you have to remember this, to counter this, in France, especially our Borodox mixture was first produced to counter this, to check this. Borodox mixture, copper sulfate and calcium hydroxide. This mixture is what we call a Borodox mixture. This can be used to check the spread of downy mildew of grapes. And the other smut is what we corn smut. As the name itself tells you, this is caused by Ustilago maydis. And unlike others, here the corn smut disease, if you observe the infected corn stem or on the corn leaves or corn cob, the infected corn, in place of seeds, here and there you will see galls or tumors, overgrowth. This overgrowth is what we call hypertrophy. So hypertrophy, hyperplasia. Increase in the cell size is called hypertrophy. Increase in cell number is called hyperplasia. Both hypertrophy and hyperplasia together make overgrowth of the given part, given area. Maybe these blisters or these galls or tumors, they can be found on the stems or on the leaves, especially in the cob, especially the grain or the kernels. Some of the kernels turn into galls. And these gall area, if you rupture it, you will see a lot number of black sooty structures of Ustilago maydis. They are chlamydospores or teleospores. So hence the smut. As the smut, unlike that, here the galls are produced as the tumors are produced. Hence, this is a tumor-like galls causing a lot of damage to the corn. Hence, it is called corn smut disease caused by Ustilago maydis. So the other is leaf spot. Of course, you read this. I need not elaborate this. Leaf spot of groundnut, whenever we say leaf spot, what is that disease called? Ticca disease. Ticca disease of groundnut caused by Cercospora personata, Cercospora arachidicola. Generally, when compared to arachidicola, the loss from Cercospora personata is very high, and generally the spots are circular in case of Personata, whereas in Arachidicula they are big and irregular, and the conidia are in Arachidicula very big, and here the conidia are small, and generally hollow, yellow hollow over the brown spots will be there in Cercospora personata. In Arachidicula, those yellow areas may be absent, but the disease or the severity or the severe loss will be through what we call Cercospora personata than when compared to Cercospora arachidicola. So the leaves, the young leaves, you will see the spots appearing, hence the Tika disease. When the spots appear in young stage, all these the leaves may become yellow in color or brown in color, or they get defoliated. So you don't find, if the entire crop gets infested with this, when the disease at high magnitude, the pods, what we call, the pods are the fruits, which is geocarpic there within the soil. So the size will be small, and even they'll be choppy. And even if the seeds are there, they're very shriveled seeds will be there. Thereby, the quality gets affected, the quantity gets affected, and farmers put to be great loss. So last one is what we call wilt of cotton. Wilt. The entire plant dries. This is what we call wilt. It is caused by Fusarium oxysporum, Fusarium moniliforme, and Fusarium moniliforme, Fusarium vasinfectum, or Fusarium oxysporum vasinfectum is the pathogen that causes wilt. Now, this wilt is because of this Fusarium produces conidia, the macroconidia and microconidia, and these conidia enter into the xylem, from epidermis into the cortex, and then finally make into the conducting tissue, especially xylem. So on entering into these xylem vessels, they move from one vessel to other vessel, and whenever the flow gets stopped, there the conidia germinate and produce mycelium. So again, mycelium, a lot number of conidia. So the mycelium, conidia, macro and micro all together, they block, they choke the xylem, conducting tissue. Hence, the flow of water and minerals to the upper parts gets blocked. So hence, drying starts from top to bottom, and due course, the entire plant dies. Hence, the name wilt. Hence, the name wilt. So in all these cases, what we have to do, so here, to check the wilt, what we have to do, cultural method, generally field sanitation should be there. You should go for crop rotation, and you go for mixed cropping will be there. You should go for what we call the wilt resistant varieties, new world cotton, such as Gossypium hirsutum and Gossypium barbadans, and Varalakshmi, Vijaya, Pratap, or Jayadhar. These are some of wilt resistant varieties that you can go for, and go for what we call pathogen free seed. So what you have to do, you treat the seed. First of all, the lint should be removed by using acids. Then mix the seed with carboxyl or carbendazim, etc. And thereby you can check, you can produce disease-free seed, and then you can go for what we call sowing. And soil solarization, fumigation, or etc. are some of the methods whereby you can check this. So in all cases, what you have to see, all the cultural methods are there, sanitation, that is cleaning and destruction of the previous crop, stubules are the infected parts. Generally what we do, what farmers do, they reap the important parts, leave the fallen leaves and the stubules, etc. So one should not do that. So they have to collect them, they have to be burnt or buried. So go for crop rotation and go for early sowing and use resistant varieties, and generally seed treatment with copper sulfate, etc., or going for Boradox mixture, spraying, etc., or systemic fungicides such as Babistine, etc., can be sprayed at a regular interval so that you can check the spread of various diseases. So this is about what we call various various fungal diseases. So we discussed about, we discussed about paddy blast disease, the rice blast. We discussed about brown leaf spot disease. We discussed about foolish seedling of rice. We discussed about black stem rust, or rust of wheat, and also loose spot of wheat we discussed. Then we discussed about what we call the important fungal diseases on millets. See, fungal diseases of millets such as sorghum, bajra, and also maize we discussed, and powdery mildews of cucurbits we discussed. We discussed about the downy mildews in case of grapes, and the ticca disease of groundnut, and wilt of cotton. So at the end, what I advise you is you remember the name of the disease and the pathogen, and the asexual mode of reproduction and the sexual mode of reproduction. Then how to control the disease, that is various cultural methods, various physical methods, various chemical methods. So the protection methods or the management methods are primarily left 3 or 4 types, especially cultural, physical, and the last one should be your chemical. So of course we had to rush through, unless and until we move, we'll not be in a position to complete this syllabus in time. That's why I had to rush through. And of course, after going home, try to read this. And if you have any doubt, you can raise them in tomorrow's class. And if you have any doubt also, now for 2 or 3 minutes, we'll try to discuss any doubt. If you have any doubt, you can raise your doubt. I'll try to clarify them. So if you have no doubt, generally, I'll call it a day, and we'll meet tomorrow at 12 o'clock, and we'll learn about bacterial, viral, and mycoplasmal diseases tomorrow. And also, we'll try to know about various, angiospermic plants, angiospermic parasitic plants, and if possible, I'll give you a brief introduction about various pests, cotton pests, and also paddy pests and storage pests, etc. Okay, no doubt. If you have no doubt, I'll call it a day, and I'll conclude my today's lecture.